Let's talk about amphibolic pathways first, and then we're going to move on to brief discussion of photosynthesis, very basic. So what is amphibolism? So amphi means both, and by drawing some amphibolic pathways, I hope I will clarify what we mean by this term. So first I want to highlight the pathway for carbohydrate metabolism. So we have polysaccharides that get digested into, for simplicity, glucose, right? C6 carbon. And glucose, during glycolysis, forms that intermediate 3-carbon phosphorylated molecule G3P, which is eventually converted as the product of glycolysis into pyruvate. Pyruvate, during the transition step, is oxidized to acetyl-CoA, which in turn enters Krebs cycle. Okay, so this is the stuff that we covered last week, nothing new here, right? So what if carbohydrates are not available in the environment? Can you use something else? The brief answer is yes, of course you can. So you start with triglycerides, and by you I mean not you personally, but bacteria. You know? So triglycerides, if you remember the formula, consist of two main categories of molecules, glycerol and fatty acids. So triglycerides get broken down into glycerol and fatty acids. Now, if the sound of the name glycerol reminds you of something that is correct, it should remind you of glyceraldehyde 3-phosphate. So glycerol, in the series of reactions, can be converted to glyceraldehyde 3-phosphate, which then can be converted to pyruvate, acetyl-CoA, and then, Krebs cycle and stuff. So far, so good? Now, fatty acids, in the process that is called beta oxidation, can be oxidized to acetyl-CoA, which also can enter the Krebs cycle. Now, what if neither triglycerides nor polysaccharides are available? In this case, proteins can be used. So proteins will be hydrolyzed to form amino acids. And then amino acids can undergo two pathways. And I'm gonna put those two pathways together because in essence, with some not so significant differences, they end up with fairly the same set of products. So those pathways are deamination and transamination. Now the results of deamination and transamination are two main molecules. So one of them is pyruvate. Okay, and you can see where it goes. And that's kind of, this is the general knowledge that I want you to possess. The products of deamination and transamination can enter the Krebs cycle. So if you remember two names, oxaloacetate and alpha-ketoglutarate, you don't need to know those details, but oxaloacetate and alpha-ketoglutarate can be the products of deamination and transamination, thus entering the Krebs cycle, continuing the metabolic process. Does that make sense? So far, like in the context of what bacteria are going to do if polysaccharides are not available or triglycerides are not available, it's going to be proteins. We good so far? What does that mean? Amphibolism. Amphi means both, and both in this context is this. What if only polysaccharides are available? Can bacteria still produce proteins? The answer is yes, of course. It can use intermediates of the Krebs cycle and pyruvate to make amino acids. Does that make sense? Can it still make triglycerides? Of course. Can use acetyl-CoA and G3P in metabolic pathways to run these reactions backwards. This is the beauty of these metabolic pathways in bacteria. Basically, many microorganisms can survive with a single source of nutrients, single source of energy, and synthesize other biological molecules from the intermediates of different biochemical pathways. That's all. That makes sense? I want to be very clear. This is enough. I'm not going to ask stuff beyond that. Any questions? No? Okay.